The International Society of Blood Transfusion recognizes 30 blood group systems with over 600 antigens. Some of these antigens are very rare and they're only found in certain ethnic groups. Others are associated with diseases or resistance to infection. And of the blood group systems, the most important are the ABO and D. There's different terms used to describe the frequency of these antigens, and you must know and be able to use these interchangeably. High incidence, public, or high frequency antigens are those present on almost every person's red blood cells. So finding blood that's negative for these antigens is very difficult. Low incidence, private, or low frequency antigens are present on very, very few individuals' red blood cells. So the advantages to both of these is the antibodies to these antigens are very uncommon, because for a high frequency, just about everybody has it, so the chances of you being negative and forming an antibody are very low. For the low frequency antigens, because so few people have it, the chances of you being exposed to it are also very, very rare. Each known blood group antigen was initially identified through the detection of its specific antibody in the serum of a patient. And that's how a lot of these blood group systems got their names. The knowledge of the serological behavior and characteristics of these blood group antibodies is critical for identification. So throughout this presentation, we're going to talk about these different systems, and at the end, I'm going to teach you a method to chunk all the material to where if you know what the antibody class is, then you know whether it's clinically significant, you know whether it causes HDFN or acute hemolytic transfusion reactions. So again, we're going to cover all this information, and it's a lot, but at the end, if you can use these simple systems for chunking the similar characteristics all together, you're going to find that answering questions on the exam or on the Board of Certification exam will be relatively easy. Antibody specificity characteristics are essential when evaluating the antibody screen and doing panel studies. Panel studies are done to identify the specificity of an unexpected antibody. Considerations are given to the phase of reactivity. Knowing the specific antibody characteristics of the different blood group antibodies are essential when evaluating antibody screen and panel studies. Considerations are given to the phase of reactivity. Is it immediate spin, 37 degrees, or Coombs, or more than one? What is the antibody class involved? If it's room temperature reacting or below, you know it's IgM. If it's 37 and or Coombs, you know it's IgG. So the phase of reactivity also lets you know the antibody class involved. Has it been shown to have the ability to cause hemolytic disease of the fetus and newborn, HDFN, or the ability to cause hemolytic transfusion reactions? This is the definition of a clinically significant antibody. So there's two classifications, broad characteristics of blood group antibodies: clinically insignificant and clinically significant. And it's crucial that you know the difference between the two. Clinically insignificant most frequently are reactive in vitro, in the test tube, at immediate spin. The antibody class is IgM, and in vivo, in the body, there's no hemolysis, no decreased red cell survival. So no hemolytic transfusion reactions, no hemolytic disease of the fetus and newborn. A clinically significant antibody is reactive in vitro in the test system at 37 and or Coombs. The antibody class is IgG, and in vivo, in the body, it has been implicated in HDFN and or HTRs. So those are the difference, and you must memorize it. And this gives you the ability, as we go along, to chunk your information. For each and every blood group system that we're going to discuss, you must know the following. Antigen development, if it's important. Antibody class, usually involved. I say usually because sometimes these antibodies don't read the books, and instead of reacting at room temperature, they react at 37. The phase of reactivity in vitro tests. Is it immediate spin? Is it 37? Is it Coombs, or more than once? What is the historical clinical significance of this particular blood group antibody? And whether donor units must be antigen negative. By that I mean taking manufacturers' typing sera and testing the donor to make certain it is negative for that particular antigen. And any unique characteristics of the blood group antigens and or antibodies, which we will discuss individually as we go through the different blood group systems. Most textbooks will divide discussion of the blood group systems into two distinct groups, the cold reacting, which are considered nuisance and their IgM class, and warm reacting, which are considered clinically significant, and the antibody class involved is IgG. If you know the phase of reactivity, you will know the antibody class involved. If you know the antibody class involved, then you will know the clinical significance. We've been discussing the I blood group system here and there throughout the course. So now here's specifically what I want you to know. It's related to the ABO and Lewis blood group systems by its biochemical structure. That's all I want you to know. At the MLS level, you would know specifically what this meant. There are two antigens, big I and little I. Fetal red cells are rich in the little I antigen, and they lack the big I antigen. The first 2 years, the I develops and they lose the little I. Anti-I reacts most strongly with adult cells because for the most part, except for the rare little I adult, all adults have the I antigen. And anti-big I will react negative or weakly with cord red cells because, as I said, the antigen develops on the red cell as the person ages. We're born little I positive, and then transition occurs to which by the in the first 2 years we transition to the adult phenotype of big I. The strength of the I antigen varies on adult cells, and rare instances of the I antigen never develops. Again, this is that rare little I adult person. So when you're doing some workups and you get varying strengths of reactions when you're doing your panel, this could be an indication if all cells are reacting but with varying strengths of reactions, 4+, 3+, 2+, you may be looking at an anti-big I. And what's important about knowing the blood group characteristics is like putting together the pieces of the puzzle. If you know the pieces of the puzzle that prove an anti-big I, then again, it's easy when you get this in a real life situation to work your way through it. Anti-big I is most frequently associated with cold agglutinin syndrome, which is an autoimmune condition. Decreased expression of big I and increased expression of little I antigens is observed in oncogenesis, thalassemias, sickle cell anemias, and it's associated with congenital cataracts in Asian populations. I antibodies are considered naturally occurring because you have them even though you've not been transfused or pregnant. They're the primary cause of non-specific agglutination in tests performed at room temperature. So with these antibodies at room temperature, you'll get positive reactions with all cells tested. Your reverse cells, antibody screen, cross matches, regardless of ABO group, because these all come from adult people. They may cause ABO discrepancies because the anti-big I, it's an autoantibody and can so heavily coat the red cells that your person will forward type as a group AB pos. When you run the control will be positive. In the reverse group, anti-big I may be of such high titer that it will react with the I antigens on the reverse cells as well as the screening cells. So again, whenever you get unexpected agglutination at room temperature, think this cold reacting antibody. In fact, eye antibodies can be detected in the serum of most normal adults if you tested their serum at 4 degrees centigrade, which is in the refrigerator, which we don't do because we don't want to see them. It's associated with atypical pneumonia caused by Mycoplasma pneumoniae, and cold agglutinin titers are used to monitor the disease. And we had a patient one time that her titers were so high. Usually in a cold agglutinin titer, you do 12 tubes with 1 through 11 is your test and number 12 is your auto control. We had to set up 24 tubes. She went that high. In fact, I was working at the hospital. I froze her serum to save for my students because I could dilute it out, and we used her forever. High titers of these antibodies may cause a hemolytic anemia. Anti-little I antibodies are rarely encountered. Anti-little I is most frequently associated with certain diseases, with infectious mononucleosis being number 1. So remember that. You see anti-little I, disease association, think infectious mononucleosis. It has also been detected in people with Epstein-Barr virus and cytomegalovirus infections. What is the clinical significance of anti-Big I? Well, it's kind of all over the place. It's usually a benign antibody that causes no problems, no issues. It's considered clinically significant in some cases of cold agglutinin syndrome. The antibodies are of a high titer, greater than 1,000. The antibody shows high thermal amplitude, which is really weird because it binds so strongly. When you have a titer greater than 1,000, and we do this room temperature and it coats the cells, then at 37, it may not come off, not because it's an IgG antibody, because the IgM has bound so strongly to the cells. And the same thing at Coombs phase, that again, the cells are agglutinated before they're washed. And I covered this in a previous lecture. It's clinically significant because it can cause hemolytic anemia. And if a blood transfusion is necessary, transfuse blood through a blood warmer. A blood warmer is a device which is hooked to the blood bag, and then the other end is hooked to the IV that goes into the patient. And it's a warming coil. It looks like a radiator, the old-fashioned radiators. And this is essential, again, for these very rare individuals who have these high titer anti-Big Is. This antibody cannot cause hemolytic disease of the fetus and newborn for 2 reasons, and you must memorize this. The antibody class is IgM, which cannot cross the placenta. And number 2, what did I say was the development of this antigen? The fetal antigen is little I. You only after birth do you start transitioning to Big I. So those are the 2 reasons anti-Big I cannot cause HDFN. Memorize that. And it does not cause hemolytic transfusion reactions, because once the blood is warmed up, the antibody dissociates from the cell in vivo. We've talked about this before, but I want to again refresh. I'm hoping by the repetitive nature of some of this information that maybe it's making more sense and maybe it will stick better. So how do you confirm an anti-Big I? You want to test the serum against 3 adult Group O RBCs and 3 Group O cord RBCs and an autocontrol. What you're going to expect is the adult cells, because they have the Big I antigen, and the autocontrol, because the patient has the Big I antigen, will be positive. The cord cells, they're either going to be negative or very weak positive. You have those overachieving babies that have already started that conversion from little I to Big I. The reactivity of this antibody is enhanced using enzyme-treated cells. And again, we talked about this in a previous lecture. They do make our panel cells pre-treated with Ficin, and Ficin enhances uptake of this antibody onto the. So this is an example of the serological conformation of anti-Big I. We have our 3 Group O cord cells. These are easy to procure if you have an obstetrical department. Otherwise, you'd have to buy reagents or just send this off for confirmation. Then we have the 3 adult cells, which are our screen cells, and then we have our auto control. These are then mixed. The serum is added and mixed, and you incubate it at 4 degrees centigrade, which is in the refrigerator. Row 1 shows reactions which confirm anti-Big I, and row 2 of the reactions indicates some other alloantibody, some other cold reacting antibody. How do I know that? Because I have negative cells all over the place. Only one of the cord cells is positive, and it's 4+, which it should be negative. And then the Group O cells, I have one adult that is negative, and the autocontrol is negative. So all of those will rule out anti-Big I as the offending antibody. Another review of the pre-warm technique, because I want you to know it, it's one of the tools we have in our toolkit. The pre-warm technique will eliminate reactivity of most examples of anti-Big I. You must use it with caution. So again, the original reactions at Coombs was 3+, 3+, 3+. And this is because if this is an anti-Big I, it's showing high thermal amplitude. So if you remember the pre-warm technique, you pre-warm the serum, you pre-warm your screen cells, you add the pre-warm to the pre-warm, incubate, and then without removing the tubes from the heat block, you add pre-warmed saline for that wash. So pre-warmed 1 is all negative. So again, this is done in conjunction with your antibody identification. You don't just do this and say, oh, I have reactions at all 3 phases. It must be an anti-Big I showing high thermal amplitude. Absolutely not. The first thing you must do is identify the antibody specificity. Yes, it's anti-Big I, then the pre-warm technique can be done, but not before, because you may end up pre-warming away an antibody that you're not supposed to. So again, the original, it was all positive, pre-warm 1. This is another piece of information that proves this was an anti-Big I. And then on the third line with pre-warm number 2, this shows that we got rid of the reactivity of anti-Big I, but there's an alloantibody underneath this autoantibody. I'm going to talk about cold autoabsorptions of anti-Big I. Very strong examples of anti-Big I may react at the Coombs test and require a cold autoabsorption, or a really cool test is the rabbit erythrocyte stroma test, which is the acronym is REST, to rule out the presence of other antibodies. Some institutions will not allow pre-warm techniques to be done because you really need to be pretty knowledgeable and have a great deal of expertise to know when you can do that test, because otherwise you may be pre-warming away antibodies that are clinically significant. So the first step is you collect EDTA blood samples from the patient and place them at 37 degrees. I need the cells, and what's really sad is if the patient does have this hemolytic anemia going on, their hemoglobin may be 8 grams hemoglobin. And so I may need 4 tubes, which is sad because they really can't give up very much blood. So we put it at 37 because this antigen-antibody reaction is reversible. So when you put this at 37, the IgM antibodies will come off the cells. Then we harvest the cells and we wash them. I wash mine at least 3 times with 37 degrees centigrade saline. Because again, the reaction, they're not coated 4+ or 5+. Sometimes these cells are coated 10+. So we need to remove as much of the autoantibody from these cells as possible. The second part is you collect serum samples from the patient. And as soon as I get that red top tube, I put it in the refrigerator. Because this way, and I lay it on its side, and allowing it to clot at 4 degrees, the autoantibody is going to go onto the cells in that red top. So I'm kind of getting rid of some of the auto anti-Big I here as well. So you spin it down, you harvest the serum. Then you add 1 ml of the serum to 1 ml of the RBCs, and you incubate at 4 degrees for 1 hour. And I do want to interject that this cannot be done on recently transfused patients because you may be absorbing out the allo along with the auto. So after the 1 hour, you spin it down, you harvest the serum, and you test against the screen cells. If the screens are now negative, then you can continue with the screen and see if it's negative or positive. If it's positive, then you repeat the absorption with the new aliquot. So absorption, auto-absorption, I'm using the patient's cells, auto, to absorb out, to get the antibody out of the serum and onto these cells. So that's what a cold autoabsorption is. And you may want to listen to this part of the presentation to really make sure you understand. The Lewis blood group system was identified in 1946 and named after the antibody maker, Mrs. Lewis. The major antigens that I want you to know about are Lewis A and Lewis B. Other antigens include Lewis C, D, and X. As an MLS, you would have to know about those, but at the MLT level, Lewis A and Lewis B are what you're expected to know. You must know about the antigenic development of Lewis. These antigens are not intrinsic to the red cells. You are not born with these antigens built onto your cells, but they're absorbed from the plasma and inserted into the red cell membrane. Is that cool or what? The genetic control resides in a single gene, the big LE. The amorph, the little LE, if homozygous, you will not have Lewis antigens. So if you're little LE, you will not have any Lewis. Lewis A is formed first, then it's modified to form Lewis B, which is absorbed preferentially over Lewis A. The Lewis phenotype of a red cell can be changed by incubating with plasma containing Lewis A or Lewis B glycolipid. So in transfusion, it matters not. This is why we want to give cross-match compatible, but let's say you have a person with anti-Lewis A, and the blood you're transfusing has Lewis A, the transfused cells will change to the Lewis phenotype of the person you're transfusing them into. Here are the Lewis phenotypes and their frequencies, and notice that there's a relatively big difference between the phenotypes for the white population versus the black population. And Lewis A-pos, B-pos is very, very rare. So the Lewis antigens are in your secretions, and they are inserted into the red cell. You're not born with Lewis antigens on your cells. So Lewis antigens in infant, the antigens are absent or extremely weak at birth. The expression of Lewis B is gradual. So at birth, you type as Lewis A, neg, B, neg. 2 months later, you'll be Lewis A, pos, B, neg. And that's where it's going to stop if you're genetically Lewis A. But most people are not. So 12 to 18 months later, you'll type as Lewis A, pos, B, pos, and because Lewis B is inserted into the cell preferentially over Lewis A, by 2 to 3 years of age, the person will be a Lewis A, neg, B, pos. Due to this reason, when we were using blood group genetics for paternity testing, you could not use the Lewis blood group system. Pregnant women can be kind of weird, and I can say that because I've had several children. And Lewis antigens in pregnancy are kind of fascinating. During pregnancy, the antigen strength may decline dramatically. In fact, many pregnant women transiently, meaning temporarily, become Lewis A, neg, B, neg. and they may produce Lewis antibodies during the pregnancy. Isn't that weird? So I'm genetically, Lewis A pos or B pos or whatever, but during pregnancy, I lose those antigens and I make the antibodies. After delivery, the antigens return and the antibodies disappear. And I don't know if it's because we've changed enhancement reagents, but this used to be a real common antibody in the serum of pregnant women, either anti-Lewis A, usually it's anti-Lewis A, but because we do things differently, I hardly ever have seen a pregnant woman with a Lewis antibody in recent years. And we talked about this, so again, a little reminder, a little review, the interactions of Lewis, secretor, and H genes. So the little LE, the little H, and the little SE genes are amorphs. They produce no detectable products. I call them placeholders to indicate the absence of those antigens. If you are little LE, you will not have Lewis antigens. But if you do have the secretor gene, you will have ABH in your secretions. If you are little SE, and have one Lewis gene, you will have Lewis A in your secretions, but no A, B, or H. So you may want to read this a couple of times just to get your brain set on what will be in your secretions based on your Lewis and secretor genotypes. If you're Lewis A pos B neg, you will be a non-secretor, but you will have Lewis A in your secretions. If you're Lewis A neg B pos, you will be a secretor of ABH, as well as Lewis A and Lewis B. For Lewis A neg B neg, 80% are secretors, 20% are non-secretors, and there will be no Lewis substances in your secretions because you are negative for both alleles. Lewis antibodies, these are naturally occurring. What that means is that you were not exposed to red cells due to a transfusion or pregnancy that had Lewis. They just happened because there are things in nature so similar that you made these antibodies. They're naturally occurring. They're not clinically significant. They're almost always IgM. They react most often at room temperature. The agglutination is relatively fragile, easily dispersed. That's why you want to be ever so careful when you shake your tubes. Lewis antibodies may cause ABO discrepancies if the reverse cells have Lewis antigens. In the past, this was a problem, but I haven't, for years, I think they've made it mandatory that our A cells and B cells, our reagent cells, are Lewis negative. They occur almost exclusively in Lewis A neg B neg people, and the production of anti-Lewis A and anti-Lewis B is not unusual. Although anti-Lewis A is the most frequently encountered, anti-Lewis B rarely encountered, and that has been my experience as well. Although most Lewis antibodies will react at room temperature, they may occasionally be seen at 37 degrees centigrade, but it's weaker and may be weakly reactive at the Coombs phase. Lewis antibodies can bind complement and cause in vitro hemolysis, and this is most often seen with enzyme-treated cells. Again, we have many tools in our toolkit when we're working up antibodies, and so there are times that maybe a patient has multiple antibodies and we want to use our enzyme-treated cells, and the person has a Lewis antibody, then those Lewis positive cells will be hemolyzed occasionally. And again, this is one more piece of the puzzle that you have Lewis antibodies present. The Lewis antibodies are not implicated in HDFN, and there's two reasons. The antibodies are IgM and cannot cross the placenta, and the antigens are poorly developed at birth. Remember, you're born Lewis A neg, B neg, and then you transition to your adult phenotype over the years. Lewis antibodies can be neutralized in vitro in our test system by the addition of commercial Lewis substance. Lewis antigens are present in the secretions, so you add the Lewis substance to a serum containing Lewis antibodies. the antibodies will be bound to the soluble Lewis antigens present in the Lewis substance, so it will neutralize them. This is useful when multiple antibodies are present and one is a Lewis. It eliminates the reactivity of the Lewis antibodies and reveals any antibodies that may be underneath or in addition to. So in row 1, we have our original reactions. We have a 2+ with screen 1, negative screen 2, positive screen 3. So we add the Lewis substance to our serum, and row 2 indicates that the antibody, the Lewis antibody, was neutralized by the Lewis substance, and there's no other antibodies present. How do I know that? They're all negative reactions. Row 3, the Lewis antibody was neutralized, but there's an additional antibody present. How do I know that? Because there are no Lewis antibodies there to react with screen 1. So the presence of a reaction with screen 1 indicates that a second additional antibody is present that you need to go and identify. For transfusion practice, the transfused red cells will acquire the Lewis phenotype of the recipient within a few days. Remember, the antigens are present in the secretion. So when those cells are infused, then they will take on the Lewis phenotype of the patient. The Lewis antibodies in the patient will be neutralized by the Lewis substance in donor plasma. Isn't that cool? So even if the donor was positive, it matters not. Those antibodies will be neutralized. Lewis antibodies rarely cause in vivo hemolysis, and it's not necessary to phenotype donors for Lewis antigens prior to transfusion, but you must give them cross-match compatible. And this patient does not meet the type and screen protocol. They're not going to be an immediate spin cross-match. You have to take that cross-match to Coombs. The P blood group system was discovered in 1927 when Landsteiner immunized rabbits with human red cells. Initially, it was just named P, but as the complexity of the P blood group system was discovered, it was renamed P1. Red cells lacking P1 are P2. There are other P phenotypes that exist, but they're very rare, less than 1%, and there are P, P1K, and P2K. Again, as an MLT, you don't need to know anything additionally about those phenotypes. And you can see that in the white and black population, there's a distinct difference in the prevalence of these antigens. When you're working this antibody up, one thing to note is the strength of the P1 antigen varies among different red cell samples, and antigen strength is reported to diminish when the red cells are stored. This is why reagent red cells have such a short shelf life. Characteristics of the antibody creates difficulties both in testing the red cells for the antigen and in the identification of the antibody. Anti-P1 blood typing reagents are usually sufficiently potent to detect weak forms of the antigen. An antibody that is weakly reactive at room temperature testing can often be shown to have anti-P1 specificity by lowering the incubation temperature or using enzyme-treated red cells. So you're doing the panel, you have varying strengths of reactions with all P1 positive cells. That's one of your clues that you're dealing with an anti-P1. The sera P2 persons commonly contain anti-P1. The antibody reacts optimally at 4 degrees centigrade. So what antibody class? IgM. But may occasionally be detected at 37. It rarely may cause in vitro hemolysis. As it's nearly always IgM, it does not cross the placenta and has not been reported to cause HDFN. And in addition, the antigen is poorly expressed on fetal cells. Anti-P1 is of little clinical significance unless it's reactive at 37. Anti-P1 has rarely been reported to cause hemolysis in vivo. There's always those unicorns. P1 antibodies can be neutralized, and one of the substances used is hydatid cyst fluid. I don't know. Some scientists are crazy. Who knew to grind up seeds and make anti-A1 lectin or anti-H lectin? Hydatid cyst fluid, this is due to an infection with a tapeworm, Echinococcus granulosus, and it has a fascinating life cycle. But unfortunately, these cysts can develop in the brain, and the fluid, once it ruptures, you're gone. So how they can grow these in vitro in a laboratory, and they harvest this fluid, and it has been found to neutralize P1 antibodies. Another source of a neutralizing substance is derived from pigeon eggs. Okay, so, are we going to crack open all these different species of eggs and figure out which ones work as neutralizing substances? I don't know, but you must memorize this. Hydatid cyst fluid or the substance from pigeon eggs will neutralize P1 antibodies. So neutralization, or another term is inhibition, is a useful aid to identify anti-P1, especially if the antibody is present in a serum with multiple antibodies. The anti-P1 is neutralized, meaning it becomes non-reactive, revealing other specificities if present. So in row 1, we have our original testing that we have positives with screens 1 and 3. We add our P1 substance, whether it's hydatid cyst fluid or pigeon egg, read the package insert, and this was a successful neutralization, so it's gone. And one thing I want to add here is the fact that we had anti-P1 identified, and now we added the P1 substance and neutralized it, and it's gone. That's an additional piece of information to confirm that you have anti-P1. The third row shows that the P1 was neutralized, but there's an additional antibody underneath. Transfusion practice for anti-P1, it's a clinically insignificant room temperature agglutinin. It's not necessary to provide antigen-negative blood. Now, you must confirm that the patient is antigen-negative, because if it turns out they're antigen-positive, then you don't have anti-P1. But donors do not have to be tested. The blood must be cross-match compatible. If the antibody is reactive at 37 or Coombs, then you would not use those units. You need to find compatible blood. And I'm so sorry this went 37 minutes. I try to keep it at 30 minutes, but again, so much to share.